Okay, good afternoon, everyone. We will start with our next session now. And I think most of you who were there on day one know that Apeksha from EnviroCare, or Apeksha is our student, ex-student, she's working in Riddle. So she wanted to introduce the EnviroCare company to you all. So, but she had a technical hitch last time, which we've solved, and she will take another 5 minutes to continue what she started on day one. So I hand over the mic to Apeksha.
Thank you so much, ma'am. Hello to all the dignitaries. So I'll be playing the video. Can we have the presentation shared? Yeah. So EnviroCare, just to repeat, it's a facility that does qualitative testing, and it's been there for 43 plus years. They've been doing this, and they have really pioneered in this. So I'll just share the video. Yeah.
Welcome to EnviroCare Labs, an epitome of excellence and innovation in the testing, inspection, and certification sector. Established in 1979 by late Shri Suresh Amritkar. Our journey began in Thane, Maharashtra, with a mission to design and execute effluent treatment plants. Evolving into EnviroCare Labs Private Limited in 1989, we marked a significant milestone in 1998 when we were recognized by the Ministry of Environment and Forest as an environmental monitoring lab. In 2004, under the visionary leadership of Dr. Nilesh Amritkar, we embarked on a transformative journey with a major World Bank project in Madagascar, catapulting us into a realm of unprecedented opportunities. As the first NABL accredited lab recognized by FSSAI for food testing services. Today, EnviroCare Lab stands as a leader in the TIC sector, recognized by reputed government and semi-government agencies. Our state-of-the-art facilities in Maharashtra and Goa transcend the traditional concept of laboratories. They are epicenters of innovation and quality. Spread over 56,000 square feet, these labs are the heart of EnviroCare Labs, where we offer an array of specialized services like food testing, water testing, environmental monitoring, pharma, and consumer products testing. In addition to these monitoring and testing services, we also provide a suite of comprehensive audit services including energy audits, water audits, environmental audits, and food safety audits. These services are tailored to help businesses and organizations enhance their operational efficiency, adhere to regulatory standards, and contribute positively to a sustainable future. EnviroCare Labs embodies the ethos of quality tested in India. This critical role in the global trade ecosystem goes beyond a mere duty. It is a profound commitment to excellence. Our rigorous testing and certification processes ensure that products not only meet but often exceed the highest international standards. CCER, Center for Continual Education and Research, is EnviroCare Lab's initiative toward quality education under SDG 4. Through our collaborations with Indira Gandhi National Open University and FSSAI, we are shaping the future of the industry. We have trained over 3,000 professionals and empowered countless students through internships, contributing significantly to the development of expertise in our field. EnviroCare Labs transcend certifications and accreditations, embodying a commitment to a sustainable future. As India's first IGBC Platinum certified laboratory building, our dedication to eco-friendliness is evident in every aspect of our operations. With 24% solar energy utilization, advanced safety systems, a wet scrubber for fume treatment, an effluent treatment plant for wastewater recycling, and over 350 air purifying plants, we ensure our environmental impact is as positive and efficient as our services. Diversity and inclusion. These are the cornerstones of our success, with a dynamic team comprising 68% women and a perfect blend of young trainees and experienced minds. We are a melting pot of ideas, innovations, and inclusivity. EnviroCare Labs believes in sustainable care. Our initiatives, such as the EnviroCare Green Awards and robust CSR activities, reflect our commitment to balancing people, the planet, and profits, driving personal social responsibility and aiming for a green balance sheet. As you journey through our facility, you are not just visiting a lab, you are experiencing a legacy of innovation, responsibility, and unwavering commitment to excellence. Welcome to EnviroCare Labs, where every test we conduct is a step towards a healthier, safer, and more sustainable world.
So that Envirocare is a partner of Riddle, where we are
Welcome.
doing this conference of Darwin, where we have facility tours, we have workshops, we have regional conferences from across other regions as well. So this is the impact so far that we have created, and this was just a glimpse of a facility that we'll be taking the team on a tour with. So yes, and anybody who's interested in Darwin, you can always contact me, and you can come to BioRiddle for any queries, and you can also scan here if you want to register right now. Yeah. Thank you so much, and thank you so much for being such a sincere audience. Thank you.
Our next session. In these sessions, total 23 students are going to present their work, and we have a limited time slot. Now we reduced our 5-minute time slot to 4 minutes. Okay, so you have to complete your presentation within 4 minutes only. Okay, so I request again, just be here near the podium, so that you can get easily on the podium, please. So we'll start with Aditi. Okay. So before start the session, the session chair, Ajaz sir, is on the way because of some inevitable work. He is joining delay. So we will start our first candidate. No, sir.
Okay. Very good afternoon, everyone. My name is Aditi Prajapati. I am from Somaya School of Basic and Applied Science. I am working under the guidance of Dr. Swati Maurya ma'am. And for the next 4 to 5 minutes, I am going to give an overview of my research topic on performing analysis and optimization of table structure recognition and text extraction. So, as we all know, tables are a fundamental component of any digital document, and pre-processing data from table is as old as analyzing the structure of the digital document. Thus, extracting and assimilating information from tabular document help the organization or an individual to make data-driven decisions, information retrieval, and many more information-related tasks. Thus, but the automatic recognition of tabular structure from the document possesses a significant challenge due to the varied range and complex layout of the table structure. Thus, this work focuses on enhancing table structure recognition in noisy and varied environment and ensure accurate text extraction, particularly in scanned document. To achieve this aim, we have outlined 3 objectives. First is to analyze existing TSR algorithms, that is to understand the advantages, disadvantages, and performing criteria of the current TSR model. TSR stands for table structure recognition. Second is to optimize detection-based model, which is basically to enhance or develop the model by identifying the current issues and applying advanced technique. And third, which is the very important, is to evaluate the algorithm performance by using the various performance metrics and to test it on the available data set. To obtain these aims, the methodology is divided into 4 stages, started by collecting the data, either from the scratch or from the public data sets available. Second stage is to detect a table from the scanned images, which is basically to know whether the table is present in the image or not. In this stage, there are various image pre-processing steps, like to convert an image into grayscale, edge detection, removing noise, and feature extraction. Next step is to recognize the table structure, which is basically to locate the rows and columns in the table. Rows and columns can be merged or unmerged. After detecting the table structure, next is to extract the text from the table, which can be done by the OCR technique, that is optical character recognition techniques, and deep learning methods. The expected outcome after following this proposed methodology, would be to offer a thorough analysis of the current TSR algorithms, thereby performing their key factors and areas for improvement. Second is to provide optimized algorithm that significantly improve the accuracy and efficiency of table detection and text extraction. Yes. That was all. Thank you. Any questions? Okay, thank you.
Thank you, Aditi. Next, we have Supriya Maurya. Dipshika Garg. Yeah, she's coming. Kiran Sharma. Please be ready, you're next.
A very good afternoon, everyone. So before I start my presentation, I would like to introduce myself. I am Dipshika Garg, and I am a graduate from National Institute of Fashion Technology, NIFT. So I spent 20 years in fashion industry. So I realized that sustainability is a major issue in fashion industry. It is the second biggest polluter in the world. So that's why I took the topic sustainability for my PhD. I'm doing PhD from KJCM. So the title of my work is Study of Motivators and Barriers for Sustainable Clothing Purchase Behavior. So in the words of Robert Swan, he says the greatest threat to our planet is the belief that someone else will save it. So fashion industry, as I said, is the second biggest polluter. So as per a report, our consumption is increasing by 400%, but so is our wastage. So the problem statement of my as I said that, we are just buying endlessly, but where is the sustainability? Who will recycle it? What will happen to our clothes if we are just keep on buying it in the name of fast fashion? So that's why when I studied, my research gap came that people there are many studies who are finding the behavior and intention gap. So I wanted to further pursue it. So my first objective is to study the motivators and barriers towards the purchase of sustainable clothing. Second objective is to understand the consumer side as why they are not able to take the action of purchase or buy sustainable clothing, even when they intend to show it that they will buy, but they are still not buying it. Next is to identify potential marketing tactics that could help reduce the intention behavior gap, thus to help industry in bridging the gap between intention to purchase and actual purchase behavior of sustainable clothing. So this is the methodology. I am doing experimental research, and this is the experimental, this is how I'm going to study. So these are the achievements till date that I'm working on 3 papers, and I have attended 10 plus FDPs. And my expected outcome is that in-depth understanding of motivators and barriers will contribute to the attitude behavior gap literature. And the study aims to enable managers to make sustainable marketing plans to help the customers in overcoming the barriers and encourage buying behavior towards the brand. This study will also be used by sustainable brands, policymakers, and government to incorporate green marketing campaigns and strategies for promoting green purchase behavior among larger sections of Indian society. Thank you. Okay, thank you.
Just because you said clothing industry is second polluter.
Yes,
large.
sir.
From the perspective waste generation or the dyes, textile dyes
Sir, overall.
overall.
Yes, sir. Like at all the stages, including the production, consumption, and selling, retailing, every stage.
So one part is complete waste generated, another part is manufacturing that
Correct,
is
sir.
dye.
Both the stages included.
So here, are you going to focus on the natural dyes or azo dyes?
So I am focusing only on the consumer behavior, intention behavior gap, that they are saying that we will buy sustainable clothing, but they end up not buying sustainable clothing. Even there is intention, but there is a behavior gap.
Okay, thank you.
Thank you, sir. Thank you, sir.
Thank you, Dipshika. Next, we have Kiran Sharma. After that, we have Shweta Sato. Are you there? Yeah.
Good afternoon, one and all. I'm Kiran Sharma, a PhD research scholar from the Department of Library Information Science, and my guide is Dr. Hindura Vaidhande sir. And topic of my research is the adoption of cloud computing in university libraries of Maharashtra, a study. Since we are going lots of transformation in the information technology, and it has affected all the aspects of our lives. So university library is also not, it has also affected the functioning of the libraries across the world. So university libraries aims is to provide the resources and services that supports to the institution's academic program and research activities. To improve the accessibility of the resources, university libraries are transforming the way they used to manage, preserve, and give access to the information sources by using different technology. So here we can say we can overcome by lots of the barriers that traditional libraries were facing by adopting the cloud-based library services. So cloud computing has several advantages to the university library, such as cost saving, quick access to information available in the library, and easy collaboration among themselves. However, adoption of and cloud computing academic library is still in infancy, and more research and awareness is needed to be explored its potential further in terms of storage, retrieval, and dissemination of the information. Therefore, this study focuses on the investigation how cloud computing has been adapted and used by the university libraries of the Maharashtra. And the study mainly aims to identify the areas of application, usage, benefits, financial requirements, technology hindrances, and issue and difficulties in adopting the cloud computing technologies in the university libraries. And the objectives of my study is actually focused mainly to answer the various questions like what is current level of cloud computing adoption in the university library across the Maharashtra, what cloud-based application and services are being utilized in these libraries, how does cloud computing affects library operations, resources, accessibility, and user experience, etc. And the main objective is to understand the cloud computing technology and its adoption in the university library in terms of the technology, infrastructure, and the awareness among the librarians and the users. To identify the cloud-based service provided by the university libraries, to explore the benefits of the cloud computing for the university libraries with respect to cost, space, and time management, to identify the challenges faced by the university libraries in adapting the cloud computing technologies, and to support and develop the comprehensive framework model for the implementing cloud computing in university libraries. For this, I have used descriptive research methodology along with the questionnaire, survey, and interview. And since we have some 86 universities across the Maharashtra, out of that, we have selected 23 universities for our sample because these universities have been NAC accredited with the A grade, and a pilot study is also will be conducted for in the Somaya Vidya Vihar University to validate the questionnaire and all. Till today, I have completed my first chapter. Second chapter is in process, and I have presented one paper in the international conference and one poster presentation in the national conference. And the main outcome of my expected outcome of this research will be the study will contribute the valuable insights into the applications of cloud computing in university libraries of Maharashtra. The research will also aims to suggest and recommend the library administration, policy makers, and the stakeholders the potential benefit and challenges after the in cloud computing adoption, etc. Thank you so much.
So I request Ejaz sir, today's session chair, to come on the dais. Thank you. Next one, please.
Next, we have Shweta Satao. After that, we have Vaishnavi Shetke. Are you there? Yeah. After her.
Good afternoon, respected dignitaries and everyone present here. My name is Shweta Satav. I am here to present my research project under the guidance of Dr. Professor Swati Maurya. The title for my research project is Optimization of Machine Learning Algorithm for Detection of Mango Leaf Diseases. India is the major producer of mangoes, contributing 50% of total world's production. It holds significant economic importance in India as it is the major exporter of mangoes, generating significant foreign exchange. It is the country's leading fruit crop, generating income for millions of farmers. So agriculture plays an important role in the economy of the country. So to improve the productivity in the agricultural field, early detection and accurate detection of plant diseases is must. So early detection and accurate detection of the plant diseases may reduce the spread of the disease, which is ultimately increasing the production and reduces the production cost. So it is it is a requirement to develop a prototype model to automate the prototype model for the process of leaf disease detection at early stage. So the proposed research, I'm using the convolutional neural network in machine learning model as it is widely used when the image data is concerned. The performance of the CNN model is based upon the which hyperparameters that we are using and their tuning also. So selecting the right combination of hyperparameters is the main objective of the proposed research, and this is the methodology you can see on the screen of the proposed research. To build a reliable data model, it is very important to have a diverse data set. So the very first step is always the data collection, then data pre-processing. So to build a reliable model, huge data set is needed and consisting, I have collected the data set of 10 different mango leaf diseases with healthy one. And this is what the methodology that I'm following. So after data collection, the data pre-processing stage is there. So whatever data set I am having, so till now I am having 4,920 images with me, which I have collected from experts from the publicly available data sets, and manually I have connected some. So to have a huge data set and to have the artificially increase the size of the data and diversity of the data set, some transformation models I'm applying to that data set, and various data augmentation techniques are used. So these are the data augmentation techniques such as geometric transformation, color space transformation, noise injection, blurring and sharpening of images and cutouts are used, which will be improving the number of the data set that I am having. After collection of the data set and the augmentation process, the very important step is to splitting the data set into training, testing and validation. So normally the proportion that is using for training, testing and validation is 70, 20 and 10. So 70% of that entire data set I'm using for training, 20% for validation and 10% for testing. So after splitting, it is given to the model training phase. After that it is compiled, and here the backpropagation and optimization training methods are used, various algorithms are used for training the model. In the proposed research, I'm using the baseline model, which is my ongoing work. I'm using the DenseNet 121 and, okay. So the basic expected outcome for my research is improving the accuracy of the disease detection, faster and more efficient processing, robust model performance, integration with the advanced technologies, and it should be usable and accessible to the farmers. Thank you so much, sir. Yes, sir.
Thank you, Shweta. Next, we have Vaishnavi Shedke. After that, we have Rohit Kumar Chaurasia. Are you there? Madhuri Kudvekar. You're next. Please be ready.
Hello everyone, good afternoon. I'm Vaishnavi Shedige. I'm a PhD scholar. I'm working under the guidance of Dr. Seema Samrani, Head Department Life Sciences. So the topic for my research was formulation of herbal nanoemulsions and validating its antimicrobial activity by microbiological methods to be used for various applications. So post-pandemic, the awareness about hand hygiene amongst the masses has increased a lot. So germicidal product like sanitizers, the demand for such products has increased a lot. But the key ingredient in this sanitizers is almost 70% of alcohol, which have harmful effects on human skin. So there are various cases of contact dermatitis, skin peeling, skin dryness, skin redness, which have been observed. Apart from this, microorganisms are gaining resistance to the alcohol that has been used as a key ingredient. There are various products that use plant extracts for sanitizers, but they are coupled with alcohol, which is used as one of the key ingredient. And the products that are developed using 100% of plant extract fail to show 90% of reduction, and there is very limited clinical studies. Also, the in silico analysis in this field is less explored. So these are some of the cases that have been registered for overuse of sanitizers. Also, the microorganisms are gaining resistance to the disinfectant ingredients. So the aim of the research was to formulate an herbal nanoemulsions which can be used in sanitizers, which will be an alternative to the chemical-based germicidal products. For this, 4 objectives were laid. First was to screen the plant extract and check its antibacterial activity. The second was to do in silico analysis of the ligands or the bioactive compounds against the target protein. Third was to formulate the herbal nanoemulsions which have antibacterial activity. And the fourth was to use this herbal nanoemulsions in the sanitizer. So for methodology, 10 plant samples were selected, and they were subjected to oxalate extraction. For oxalate extraction, 3 solvents were used, and in all 30 extractions were carried out. All the antibacterial activity of all these 30 extracts were checked by well diffusion assay, and the one which showed maximum activity was shortlisted. So clove and cinnamon and rosemary and hibiscus were the extract that showed best antibacterial activity, and to identify the bioactive compounds present in them, GCMS analysis was done, and the major bioactive compounds were screened for. For molecular docking, 3 softwares were used, that is Autodoc Vina, Autodoc MGL tool, and Pymol visualization tool. To check the safety of the bioactive compounds that are present in the extract, ADMET analysis was carried out. Since clove and cinnamon oil showed best antibacterial activity against all the selected microorganism, and it also showed highest antibacterial activity, so they were selected for nanoemulsions. So nanoemulsions were formulated by herbal nanoemulsions. Particle size was determined. The antibacterial activity of those nanoemulsions was carried out individually and in combinations, and the effect of those sanitizer on skin human skin cell line was also studied. So these are the results of all 30 extracts against 8 bacterial species. This was some of the bioactive compounds that were detected in GCMS analysis. The highlighted one are the one which is less explored bioactive compounds and showed best molecular docking studies. So this is a molecular docking which was one of the significant, which showed interaction similar to the cefotaxime antibiotic. This were the nanoemulsions of clove and cinnamon. This was the antibacterial activity, where we can see the last two plates from the each plate showed no antibacterial activity. Just 2 minutes. This is the sanitizer. It is its efficiency. Also, it falls under the nano ranges. So this was a study on the skin cell line where it followed the ISO standards. Also, there are no morphological changes in the given cells, so it can be regarded as safe as ISO standards. These are some of the publications of which 3 are already been published and 1 is in the accepted and the communicated format. These are the conferences where I have presented, and one of the national symposium where I have bagged first prizes. Thank you. Questions? Thank you. Yes, sir.
To monitor that as well.
I have only done, what I have done was SRB assay, wherein the protein of the cell was studied. So if the cell is healthy, in healthy state, so it can be regarded as safe.
All right.
So in that way, I have done protein study.
Okay, yeah.
Yes, ma'am.
Which are the bacterial strains that you actually looked for the antiviral, antimicrobial activity?
Ma'am, 8 bacterial strain: E. coli, Aureus, Salmonella, Shigella, Aspergillus, Proteus, Klebsiella, Bacillus.
So these are normally using the skin cells as their host?
Yes,
Or
ma'am.
are they
Of this,
living on
staph and strepto are the one which are which are responsible for skin infections.
Thanks.
Thank you.
Thank you.
Thank you, Vaishnavi. Next, we have Madhuri Kudvelkar. And after that, we have Tikaram Ganpat Verma. Tikaram, are you there? Okay.
Good afternoon to all. I'm sorry I'm not adding my institute name because the PPT was made on time for the submission. My name is Madhuri Kudvalkar. I'm a PhD student of KJ Somaya Dharma Studies, KJ Somaya Institute of Dharma Studies, and I'm doing PhD in Buddhist Studies under the guidance of Dr. Pallavi Nalavade Zamde. My research topic is the title work is Study of Present Status of Buddhist Bhikkhuni Sangha in Maharashtra, and the importance the current research is required to deal with the spiritual, social, and cultural denotion of Buddhist Bhikkhuni Maharashtra. In Maharashtra, the qualitative and the quantitative outcomes of this research will contribute to plan, policy recommendation, and academic discussions and Sangha community uplifting activism. Problem statement and objectives: to study the present status and conditions of the Bhikkhuni Sangha situated in the different parts of Maharashtra and try to find the solutions for their betterment. Objectives: documentation of existing Buddhist Bhikkhuni and Sanghas in Maharashtra, then an understanding of the problems of Buddhist Bhikkhuni and the Sangha, then a study of the present status of Buddhist Bhikkhuni and their Sangha in Maharashtra. The methodology which is planned: the survey method has been selected as the primary approach for the research. Then the data collection tools are observation, that is direct observation will be conducted to gather insights about the daily lives, practices, and the interactions of the Buddhist Bhikkhuni Sangha, in-depth interviews with the key respondents, including bhikkhunis and the community members, will be carried out to capture the personal narratives and perspectives. Structure questionnaires will be distributed to gather quantitative and qualitative data on various aspects of Bhikkhuni Sangha's conditions. Fieldwork will be also there. So fieldwork will involve visiting relevant sites associated with the Bhikkhuni Sangha to observe their living conditions and practices, conducting meetings with the respondents to establish rapport and facilitate open discussions. The achievement till date and expected outcomes. Achievement till date, the research is ongoing. The research has made the existing Bhikkhuni Sangha and it took interviews among Bhikkhunis. Researcher documented about the problems like ordination, seminary training, dana problems, conflicts, delays in the constructions of Bhikkhuni Vihara, and upcoming challenges. Researchers also attended the International Sangha Day with the Bhikkhuni Sangha in Vashi and Bangalore to observe the activities and organization. Till now, researcher has documented about 100 bhikkhunis. The expected outcomes will be, upon completing this research, the conclusion will help establish goals for addressing the challenges faced by the Bhikkhuni Sangha. The recommendations outcomes will inform policy development aimed in improving the conditions of the Bhikkhuni Sangha. Both qualitative and quantitative data will be provided for future research studies. Thank you.
Thank you, ma'am. Questions? We'll take one if there is. I'll take one, ma'am.
Okay, sir.
कितने टाइम से कर रहे हो आप? साल हो गए?
DSC has approved my topic in May 23, and the research is just started.
Just started. Okay, thank you.
Thank you, sir.
Thank you, Mayuri. Next, we have Tikaram Verma. After that, we have Nikhil Desai. Nikhil Desai, are you there? Ketki Joshi, you are next after.
Good afternoon all. I am Tikaram Verma. I am the PhD scholar in Mechanical Department of KJ Somaiya School of Engineering. So currently I am doing my PhD work under the guidance of Dr. Shailesh Nikam sir. My title of work is Aerodynamic Evaluation of Bluff Body. So as you can see in the figure, it is very important to evaluate aerodynamic behavior of a bluff body whenever the flow is happening across a bluff body. At the back side of the bluff body, there is a continuous vortex shading that causes a drag force on the bluff body. This creates a lot of problem. So the importance is to understand the vortex shading, its phenomena, and because of that, because of the vortex shading, a lot of forces are being applied. On the bluff body. So we need to reduce that forces. At the same time, if we can reduce the forces, we can do energy efficiency calculations, and then all this parameter will going to affect the safety design of the transportation vehicle and various other parameters. If you recall, there was some incident some 5-6 months back. There was a huge hoarding which was collapsed in the Mumbai. So the main reason was the vortex shading only. The problem statement is to investigate various bluff body forces. The objective is to investigation of vortex shading behind bluff body and the effect of Reynolds number on the aerodynamic forces. So this is the experimental facility we have in the Bhaskaracharya building. We have a wind tunnel, which is the capacity of the wind tunnel is from 0 to 30 meter per second, and the test section is 600 mm by 600 mm. So currently I am conducting an experiment in the bluff body of circular shape of 50 mm dia. You can see in the figure. So this experimentation is going on. I am conducting the test at various velocity, starting from 5 meter per second to 30 meter per second. These are the results of the experimentation. So from the experimentation, I have calculated a CP, that is a pressure coefficient, and I have plotted the CP versus theta graph. So the graph shows the separation for the laminar flow occurs around 80 degree. And in this graph, I have also included some of the flow which are from turbulent zone, and for the turbulent zone, the separation occurs at 120 degree. So the main focus of attention is here. When the separation occurs earlier, it creates a lot of low pressure in the backside that causes a lot of drag force on a bluff body. So as a experimentation, I need to focus my main aim is to find out at what point I can delay the separation. If the separation is delayed, that will be good for the setup, and the less drag force will be acted on the bluff body. So the CP versus theta graph is there, and the another graph I have made is CD versus Reynolds number graph. So for my work, this work is carried out at 77,000 Reynolds number. So in the facility which are having in the winter in our lab, the capacity is from 3,000 to 90,000. So beyond 90,000 we cannot test. So I have conducted all my tests in the range of 3,000 to 90,000 Reynolds number, and I have found that the CD is more or less same throughout this range. Expected outcome. So I have found out that the CD value doesn't change throughout the Reynolds number from 3,000 to 90,000, and the coefficient of pressure plots helps to identify the separation point, which is 80 degree for the laminar flow. Thank you.
Thank you.
Thank you, Tikaram. Next, we have Ketki Joshi. After that, we have Afreen Shah. Afreen, are you there? Yunus Dalal. Ashwini Bore. After her.
Good afternoon to everyone present here. My name is Ketki Joshi and I'm an undergraduate TY student, and this is my presentation on my research project that I've been working on. And the topic is From Solvents to Strategies, Harnessing Lacase Production in Solvent-Tolerant Bacteria. For this project, I have been guided by Dr. Seema Sambrani. So let me tell you about what are organic solvent-tolerant bacteria. These are bacteria that have developed adaptations to survive in toxic environments that have been created by the presence of solvents such as benzene, acetone, and toluene. But the fact that they can survive in this kind of environment shows that they have some special kick to them, that they can survive in this, plus they must have some robust enzymes and other metabolites that we could extract from them and use them for our purposes and our industrial applications. So there is great environmental potential for this enzyme known as lacase. It is a oxidoreductase. And as you can see, I've put the structure over there. It has two catalytic centers. And in short, the best part about this enzyme is that it breaks down complex and hazardous substances into almost toxic products. Mostly it converts them into water and oxygen. So the aim of my study was to obtain solvent-tolerant bacteria from natural sources and to study their enzyme activity. So these are my objectives. Of these, I have completed the first two, and in this semester, I plan to complete the other two. This is my methodology. I have taken a soil sample from near a petrol station and a marine water sample. Then I slowly exposed it to a solvent known as toluene, and I isolated the bacteria. Further, the plate that you see here, the orange plate, the reddish coloration is formed when there is enzyme that is being produced by that bacteria because it is breaking down a substrate that I have added to that plate known as guaiacol, and that gives that reddish brown color. Further, I have allowed the bacteria to grow, and I have used the crude enzyme extract to observe how it breaks down the substrate, and I have taken the absorbance of that at 470 nanometer. The interesting thing here is for checking the enzyme activity, in one set, I checked the crude enzyme with the substrate alone. But in the second set, I added the solvent toluene into the reaction mixture to see if the enzyme was able to work or not. And this on the side is my upcoming work where I'll be extracting purified enzyme and using it for the dye degradation studies. So these are my 8 isolated cultures. Of these, 2 are gram negative and 6 are gram positive. And as you can see, the screening shows the 8 cultures. The orange zone that you see shows lacase activity. These are my results, and these are the parameters that I have checked. So I have checked pH, temperature, agitation, carbon source, nitrogen source, and salt source. Of these, I have written which all I assayed, and on this side is written which one was found to be optimal for most of the cultures. And most interestingly, one of my hypotheses when I first started the project was that I hope to find bacteria that would either tolerate or get enhanced activity in the presence of solvent. And for some of them, that was not the case. But for one of them, it showed heightened activity in the presence of solvent. And I'm very excited about taking that further. Sorry. So I presented my research work at SVKM's Dr. Bhanuben Nanavati College of Pharmacy in Vile Parle. And I received the first place for my oral presentation there in the UG category. So these are my expected outcomes. Shall I conclude? Yeah. So so this is just what I intend to complete in the next few months. And studies show that this is the molecular weight of lacase between 55 to 70 kilodaltons. So I hope to get the same result in my studies so that I can effectively confirm it. Thank you.
Bacteria are extremophiles, or they
yes, sir,
you can call them.
Yes,
like
sir, because they were isolated from near a petrol pump. So the fact that they can survive there means that they have some extra adaptations. So they can be called extremophiles.
Yeah. Another parameter what you can put in your experiment is like spinning them at very high, rotational velocities. So people have studied this kind of like, what kind of gravity they can withstand, like, extreme pressure conditions. So that you can easily simulate in a lab centrifuge.
Right, sir.
Yeah. So you could add that as another dimension.
Yes, sir, I'll do that.
Yeah, thank you.
Thank you, sir.
So we are very happy that undergraduate student is presenting with so much confidence the research. What is your future plan after your completion of graduation?
Sir, I have been interested in plant biotechnology since I was in second year, but now that has translated into environmental, especially through this project. And this is then taking me to healthcare, so I might go into biomedical science.
You want to do PG?
Yeah, for PG. For
Not
my
going
MS.
for job?
I believe I will do a dissertation master's, and then I will decide, because I enjoy research very much, but I would like to, I'm equally between industry and a PhD, per se.
Do you read the journal articles related to your research?
Yes, most definitely.
Your guide is giving or you are downloading?
Both, I mean, we share, she keeps sending me things.
Good. All the best.
Thank you, sir. Thank you.
Thank you, Ketki. Next, we have Ashwini Bhore. After that, we have Gaurang Chetty. Gaurang, are you there? Himanshu Mishra, are you there? You're next.
Good afternoon all. Myself Ashwini Bhore. I am pursuing my PhD under Dr. Hindurao Vaidande, University Librarian, Department of Library and Information Sciences, Somaiya Vidyavihar University. My topic of research is use of digital tools in scholarly publications, a survey. As we know, the reputation of every country, every university depends upon the quality of research outcome published by that particular university. And nowadays, ICT offer various digital tools to write a research publications. Also, because of the mandatory mandation of research to publish a research article for NIFR ranking, for UGC NAC, for career advancement skills, and as a part of research work, researcher has to publish a research articles, but if they have implemented tools in their writing a research paper, they can enhance the accuracy and credibility of their research work. So, and in my day-to-day life, I observe as a librarian, many of people face the problem of research paper rejections, and they. So, I have choose this topic for my particular study. The objective of study is to study digital tools in scholarly publications, to describe utilization pattern of digital tools by researchers, to measure the impact of digital tools on quality of journal publications, and to identify the factors responsible for its minimum usage in the digital environment, and to make appropriate recommendations based upon the finding of the study. Hypotheses are: using digital tools increase the quality of research publications. Implementation of digital tool in research writing increase the citations. Using digital tool in scholarly publication increase the possibility of acceptance in high impact factor journals. So, methodology plan for this particular study is descriptive quantitative research method with survey technique, and the tool for data collection is 5-point Likert scale questionnaire, one for librarians, and one second questionnaire is for the research student and research faculty. Population selected for this particular study is major 25 research institutions in Mumbai, and data analysis through SPS software which will be interpreted by the bar charts for analysis and conclusion. So currently I have completed my introduction chapter. Second chapter, literature review, is under a progress. First questionnaire for librarian completed as a pilot for pilot study, and second questionnaire for the researchers to collect the data on research awareness and purpose of the use of the digital tools and frequency usage of the digital tools. Researchers gone through for the collection of the population, researchers gone through the website of the research organizations. Meanwhile, I have presented 2 papers, one in a national conference, one in the international conference, and one poster presentation. The proposed outcome of this particular study is use of digital tool in writing scholarly journal publication, improve the reliability, accuracy, and ultimately enhance the quality of research publication, thereby increasing the acceptance in the high impact factor journals. Future endeavors should be focused on the AI-based digital research tool in scholarly publications, and adoption of these particular tools in writing a research aids the process of research writing. Thank you.
Thank you, Ashwini. Next, we have Himanshu Mishra. After that, we have Sanskruti Jagadesh. Are you there? Okay, you're next.
Good afternoon, everyone. This is Himanshu Mishra. I've just got inducted into PhD, and though the topic is not yet finalized, is not stamped, but basis the discussion what I have so far with my guide, Dr. Ketan, we are going to evolve around this topic only. So the topic is examining the impact of ESG integration into listed corporates and the long-term and the impact on the financial numbers and the long-term sustainability of the organizations. The extension of the research or the main focus would be around the, out of the ESG, environment, social governance, the main focus would be on the governance, because that is the main pillar which impacts E and S as well. So the importance of ESG, which is gaining momentum at the moment in India, 2012, when BRR was made mandatory by SEBI for top 100 institutions, listed corporates, and then 2021, that was made the change in the BRR process, which made BRSR, which was introduced in 2021 for top 500, and right now which is there for 1,000 corporates. Why I'm mentioning this? Because the gap which is there at the moment on this research area is that we don't have a very long or data which is available. So all the researchers which are there are mainly on the short-term impact or of ESG integration. If we need really to work upon, though it's a mixed approach on data collation, the dependability which is there on the BRR, whereas BRSR, the technical terms where the earlier version where the data which was being seeked by SEBI was mainly on E and S, whereas 21, then the entire questionnaire which was there of 50 questions which made to 140 odd questions now by SEBI, which is more elaborated and having a more concentrated focus on the governance. And because my intention is to work on governance, the data is going to be a real big challenge. The importance of the topic where the investors or the regulatory pressure which is there or the risk management or the enhanced reputation of the organizations who have the higher ESG rating. And when I say ESG rating, there is no standard format or a framework which is being adopted by all these agencies who rate them. So there is another lack which is there, because if you are comparing the two ratings of two different agencies, you cannot be sure that we cannot compare one rating to other rating, because the framework is different. The problem statement, which is, as I mentioned, the gap which is there, a lot of studies. One of the best study what I had is by Dr. Khan, which is more on the approach on or the ESG integration impact on materiality especially. But all the researchers, whatever I have read so far, mainly focuses on the short term and not on the long term impact. And now, because the data which is there, not enough data which is there in the domain, Bloomberg also stopped sharing the data. Thomson Reuters is having up to some extent now. I really need to have the access of that data as well, where the Indian corporates data is there to have access of that. So in the mixed approach, one is data, but because I lead ESG for my institution, and then I might have approach to other ESG leads in the industry. So I have to rely upon the qualitative data as well to a large extent. And the expected outcome, especially my idea, is to have some impact on the regulation side. If I can impress the regulator that the governance is one of the major pillar which is having the impact on the sustainability of the organization, and not sustainability as a word sustainability when we speak on ESG. Thank you.
Any question?
What is the structure of your data? What is the expected data for your analysis?
So when we speak on quantitative data, the current framework which is there from 2021 onwards, we are having a reporting on the annual reports of the listed corporates of top 500 to start with, and now it is there for top 1,000 corporates. There the data is there on the absolute numbers. There we can have return on assets and other financial numbers. And we have a 140, which is also divided into 98 and 42. One is essential data, which is there for all the corporates from last 4 years. But there is still another 42 questions which are optional by the corporates. So there's no standard framework which is there. So the data which is required to assess the impact of the ESG integration. And another challenge which is there, that, because we cannot have a constant P for all the corporates, and only with the variable of ESG, how can we correlate both the numbers that the impact of ESG is there in the long-term number of financial number of the organization? That is going to be one of the challenge. And
okay, we can talk offline.
Yeah, sure, sir.
Mr. Mishra?
Yep. Sorry.
No, I just want to know that you're struggling with finding the data where people are not very open about it. But listed companies do have accessible, right? Like you can access them.
Yeah. So that's what I'm saying. Earlier, the entire data was there at the Bloomberg, but there also, if you are having data only for the top 100 corporates, which is there from 2012 12 onwards. But you have to dependent on top 100 data only, and if you want to do some research on these industry specific data, you might not have enough sample to that, but we'll find ways and means
Tying
to do.
up with NSC might help.
Yeah, correct. Right, sir.
Thank you.
Thank you so much. Thanks.
Thank you, Himanshu. Next, we have Sanskruti Jagadesh, after which we have Sanika Kinkar. Sanika, are you there? Yeah.
I see.
Okay, a very good afternoon to everyone present here. I am Sanskriti Jagdesh. I am currently a third year undergraduate student pursuing my biotechnology degree. My guide is Dr. Seema Sambrani, and the title of my project is Effect of Magnetic Field on the Growth of Spirulina. I'm very glad to announce that this is a Venyan scholarship funded project. Now, microalgae like spirulina, they are known to absorb the atmospheric carbon dioxide and convert it into oxygen and biomass. But what makes them so attractive is that they have the ability to produce a lot of valuable compounds that are of commercial interest. And spirulina especially has a high amount of protein content in its nutritional profile, which makes it very appealing to the health conscious community. Now, since there is a need in supply, since there is a need in demand, there's also a need in supply. And we need better and non-conventional ways to grow spirulina at a larger scales in a better manner. Now, magnetic field is known to induce its or have its effect in microalgae like spirulina with respect to its growth, its nutritional profile, the secondary metabolites that these organisms produce. And there is a dire need to optimize the magnetic intensity, the duration of the whole study, and that is what I aim to focus on. Now, I wanted to use waste plastic 2-liter cold drink bottles as a source of bubble column photobioreactors, and bubble column photobioreactors are also known to be a very simple and efficient way of culturing these microalgae, as they're very easy to use in a lab scale level. Yeah, I'm pressing. Yeah. So the objectives of my study were to first culture or start my mother culture, which I can use as a source of inoculum for my control and test studies. And the next is to perform a comparative study where I'll be comparing a control system under no magnetic influence with test systems under magnetic influence to see the difference. And the third one is to perform a broad nutritional analysis covering the aspects of carbohydrate, protein, lipid, chlorophyll, carotenoid, and phycobiliproteins, and also RNA estimation to see what the magnetic effect does. Now, this is the brief methodology which I have employed till now, where I used the live starter culture to make my mother culture. Then I used it to set up my control systems, and the harvested biomass was used for the nutritional analysis. And the results that I have achieved till now are displayed here. I have seen an increase in growth with respect to increase in magnetic effect as well. And proteins and carbohydrates level have also increased with the induction of magnetic field. But the other factors, I think I need to perform a duplicate study to get more conclusive results. Now, my ultimate outcome or aim is to have like a set guideline to ensure that there is increase in the growth of my spirulina under the influence of magnetic field, and to optimize which parameters have to be used for the same.
And like I said, I'm a proud recipient of the Venyan Scholarship in the Bachelor's Project category. And the scholarship awarded to me was of 10,000 for 3 months. And I've already received the first 2 installments, and I'll be receiving the last one once my project deliverables are fulfilled. Thank you.
Good, congratulations for receiving this scholarship.
Thank you.
This is you have to submit some expenditure of this bills, anything
So,
to back to
no,
them?
sir. For bachelors, they have given full freedom. I can utilize the funds any way I want. But they have every month assessment sessions where they do keep
Okay.
track of the
Progress.
students'
That
progress.
is okay.
Yes.
So you are utilizing that 30,000 for
Project. Till now, I have spent around like 5,000 on buying magnets, the bottles, and also this system requires me to buy like a fishing tank pump for aeration. All that I have utilized from the project's fund to purchase all of that.
So any journal article is planned for this work?
Yes, so this project is only of 3 duration, but I am continuing this for 6 months for my internal thesis, and after that, along with Seema ma'am, we'll share it.
Is it journal identified?
Not yet, but I am looking at side by
Identify.
side. Yes, yes.
Because start reading the papers in those journal and start working on those lines, then only
Yes.
it will be accepted.
Yes. Thank you.
There's 2 questions to ask. No, why you wanted to study effect of magnetic effect on the growth or so?
So why especially magnet, I do not have a very clear answer, but the reason I chose this topic is when I was doing my literature review and choosing a topic for my proposal, I found that magnetic effect, when it was induced in other organisms, it did show a positive correlation with respect to growth, and I did not find many papers for spirulina. And at the same time, I came across another article which said spirulina is the it topic these days for as a health supplement or a nutritional supplement. So I thought, why not apply magnetic field on spirulina to see?
What is the core of the chlorophyll?
It is magnesium.
So magnesium is ferromagnetic in property, right? And magnesium is again core factor for many enzymes.
Yes, yes, cofactor.
Enhancing the activity by magnetic field might be helping increase the activity enzyme, and definitely production will increase.
This topic
so this
has
is a macro or microalgae?
This is microalgae. Spirulina is microalgae.
Are you sure? Not macro.
Yeah.
And all the graphs you have shown, no, there should be standard error bar. You have to repeat some of the experiments.
Yes, sir.
As I mentioned, we want to go for publications.
Yes.
So you have to repeat some experiments. There should be standard error bars on all the graphs.
Okay, sir.
Right. Thank you.
Yeah, no, I was about to ask the similar thing only, like, what is behind the increased growth, what you are seeing?
The exact mechanisms of how exactly the magnetic field does this is not.
So one possibility is something like what is proposed, and it makes good sense that since you want to cultivate this algae much faster, and if magnetic field could increase the rate of growth, it's a wonderful thing.
Yes.
It's a very valuable result. Yeah, thank you.
Very valid point. Thank you.
Thank you, Sanskruti. Next, we have Sanika Kinkar. After her, we have Amit Narwade. Amit, are you there? Next.
Good afternoon, everyone. My name is... Good afternoon, everyone. My name is Sanika Nilesh Kinikar. I'm from TY BSc Biotechnology Honors. I'm very grateful to say that I am an awardee of Venyan Scholarship and Mentorship Program, which is organized by PKC Pune Knowledge Cluster with the collaboration of VSF Chemicals, India Private Limited. And for that, I'm receiving 10,000 rupees per month for 3 months. Today, It's an absolute honor to stand here and present my research topic on bacterial degradation of benzyl chloride, a project that symbolizes the fusion between microbiology, industrial pollutant control, and environmental sustainability. Now, imagine a chemical which is widely used in industries to produce dyes, perfumes, and even pharmaceutical. But it is also so hazardous to air, water, and soil. So, even to human health and ecosystem, this chemical is benzyl chloride. Now, the traditional ways to remove benzyl chloride often involves the treatment of chemicals, which are expensive, and it also gives some secondary pollutants to the environment. As being a biotechnology student, we thought that there is a much better way to use nature, which shows a better alternative for this, like microorganisms, particularly using bacteria with the potential to degrade such toxic compounds into harmless byproducts. So, my primary objectives for my research are to isolate solvent-tolerant bacteria which are capable for degrading benzyl chloride, to investigate the degradation pathway of benzyl chloride by these bacteria, and to enhance efficiency of using microbial consortia. Now, this is my methodology. To find this remarkable bacteria, I collected the soil sample from petrol pump, a site which is rich in solvent-tolerant microbes. And then using serial dilution and spread plate technique, I isolated bacterial strains that are exposed them to benzyl chloride. Then this step created selective pressure, ensuring that the most resilient and degradative microbes should be survived. The bacterial isolates were inoculated with benzyl chloride and toluene for comparative studies, which set the stage for my crucial part of research, doing metabolic analysis. So far, the isolates that I've obtained are gram-positive and shows very promising degradative potential. The upcoming GC analysis and Rothera test will provide a deeper insights into the efficiency and preferred degradation pathway. So, my expected outcomes are GC analysis, gas chromatography. This will help me to detect the intermediate compounds formed during the degradation. Some expected metabolites include benzyl alcohol, benzyl aldehyde, benzoic acid, and catechols. Then the next study I'll be doing is using Rothera's test. This will confirm the presence of ketone intermediates. If a positive result comes, it will indicate it by deep purple magenta color. Will validate the cleavage of benzyl chloride, and it will indicate whether the benzene ring has undergone meta or ortho cleavage. If it has gone through ortho cleavage, it will lead to cis muconic acids, which will be further can be metabolized into TCA cycles. If it undergoes meta cleavage, it will result in 2-hydroxy muconic semialdehyde, which follows a different degradation pathway. Now, these insights will help us to understand how these bacteria can naturally detoxify benzyl chloride and how we can enhance the process for large-scale environmental applications. Moving forward, the goal is to optimize microbial consortia, ensuring faster and more effective benzyl chloride degradation. Thank you.
And all of three, you have being undergraduate, doing great job, and we want many more students for undergraduate and postgraduate level should take up small research projects and do such work. So further refinement may be done with the help of your teachers and guide. Like normally bacteria, when they get exposed to any toxic compound, they try to adjust themselves to toxic environment by two ways, either by oxidizing that compound or by reducing that compound to making it less toxic. And they can use this as a hydrocarbon source of energy. So what kind of mechanism you are looking in your organism?
I'm looking like if they are degrading, so I'll be checking the byproducts they're making, and they'll using benzyl chloride as the main source of carbon. They'll be using benzyl chloride and toluene for the main source of carbon,
so you have to
nutrients
do a lot of
and
enzyme assays.
Yeah.
Like some of the students, he's doing already lacase.
Oh.
Now one of the students is doing.
Yeah.
So you have to do lacase, hydrogen peroxide, peroxidase, oxidoreductase. All these enzyme assays you have to do. One of them you will find, and then go for which are the reduction products. That where you have to use for the GCMS.
Yes, sir.
And purify the reduced, reduced products separately with the enzyme activity. Then you can develop the pathway.
Yes, sir.
of the reduction.
Yes, sir.
So after identify 2-3 more organisms, you can develop the consortium to enhance the further level to
Yes.
reduce.
Yes, sir.
So plan it for next your MS program also.
Yeah, I have my internal thesis for that. This is just for 3 months, so these are my finance objective.
Right. So good, good, great. Congratulations of teacher also and students also.
Thank you, sir.
Sir, can you pass the mic?
So yeah, when I heard benzyl chloride, the immediate thing came to my mind was like benzyl hexachloride also was there quite in use, like BHC, right? Have you heard about this compound, BHC?
No, sir.
I think maybe perhaps it's banned nowadays. Earlier, like, yeah, okay, it's recently banned then. So this used to be available in all the households in villages, so because this is an insecticide, and it has caused severe harm, and the government has banned it recently. So it's very interesting that bacteria can actually decompose. A very interesting problem. And along the lines, what he was suggesting, like if the bacteria could have, if the carbon consumption is the idea, then maybe you can use BHC also if it's available, like as a kind of a comparison, like whether BHC has lesser carbon, more chlorine.
Yes,
so whether there is a preference over these two. So there are many things you can push in the very interesting problem.
Sure,
yeah.
sir. Sure.
Thank you. Yeah.
Thank you so much, sir.
Congratulations and thank you. You
Thank
are
you, sir.
doing a good work at the UG level.
Thank you so much, sir.
Keep that spirit.
Yes, sir.
And do something. On the first day, I think our Vice Chancellor sir said that you were there in the hall?
Yes, yes.
He said that undergraduate students are publishing in which journal, he said?
That I don't remember.
Nature,
Achha,
Science.
sorry.
So we look forward to students like you to contribute to such journals with the help of the senior faculty members, because it is not going to be easy task there. All the best.
Yeah, I also just add a comment, like, many of you are collecting samples from the petrol pumps. I think Mumbai petrol pumps are supporting, research quite a lot. They are quite generous, looks like. Yeah, thank you.
Thank you so much, sir. Thank you.
Thank you. Sir, but petrol pump, they don't have any soil area.
Next we have
it's all
Amit.
concretized.
Next we have Amit Narwade, after which we have Akash Bidwait. Akash, are you there? Rohini Nair. Poonam, Miss Poonam. Radha, Miss Radha. After.
Good afternoon to all. Myself, Amit Narwade. I am basically from the engineering background, but afterwards in master degree, I have choose humanity as a subject. And I am pursuing my PhD in KJ Somaya Institution of Dharma Study. And my guide, Madam Dr. Bhagyashree Bavre. Here, before starting with my presentation, I would like to tell you about the achievement till the date. The DSC meeting has recently accepted the research topic and my research proposal. I started with the literature review, and primarily I finished my introduction part. And the literature review with the different scholars I have started with. Recently, I have presented a research paper at an international student conference, Savitribai Phule, Pune University, on the subject of the role of Buddhism in social conflict resolution. So the research topic is basically the study of Anagarik Dharmapala's literary work to analyze his perspective on the Buddhism. Basically, many of the people in this hall may not be aware about the Anagarik Dharmapalas. So I just wanted to give a very brief introduction. He is uh, one of the personality for the revival of the Buddhist movement in colonial period. So the importance of this study is the study of Anagarik Dharmapala's literary work to analyze his perspective on Buddhism, intended to study the Anagarika's scholarly work and his thought about Buddhism from various literary sources which are published in various journals, books written by him. Anagarika's movement for the revival of the Buddhism was backed up with his strong conviction in Buddhist philosophy, his interpretation of the Buddhism, and also in his understanding of the Buddha and his teaching. The problem statement is basically, it is found that many scholars have studied and emphasizes on the Anagarik Dharmapala's struggle to revival of the Buddhism and its movement in India, across the globe as well. However, they have not analyzes the philosophy of Buddhism from Anagarika's perspective, which is expressed through his literary work. Here are some objectives I have identified to study the Anagarika's literary work to analyze his ideological foundation of the Buddhism, which is helping to build his Buddhist revival movement in India and globe. Second, the study and analyze his perspective on the decline of the Buddhism in India, and also to assess his contribution to Buddhist literature. Here are some, hypotheses I built to carry on the research. The revival of movement of Anagarika developed as a religious movement, and the ideology expressed in his writings also reflect the same, focusing less on the contemporary social systems in India. Anagarik Dharmapal present a very different perspective on certain concepts in his writing, primarily associated with the religion. The methodology which I have adopted here is the source material for analysis consists of literary work, interviews, video recordings, photographs, writing works, etc. In this journey, I'll move very fast. The approaches which I have considered here is the historical approach and the philosophical approach, wherein the historical approach, researcher has adopted a study the philosophical foundation of Anagarika through his literary work. The theories which I have involved in this research are post-structuralism and post-colonial theories. So this is how I am going to identify the literary work of Anagarikas. Thank you so much. You can ask if the questions are any.
I have not specific question, but it's very good that you are an engineer and then gone to Dharma.
Thank you, sir. Thank you.
Typical good example of interdisciplinary.
Yeah.
Transdisciplinary.
Yeah, thank you, sir.
Secondly, as you said, Buddhism is reducing in India. So do you think is over time from hundreds and thousands of years, there is some new religion will come, rise, go to the peak and go down? So it is the process of cycle is there?
No, let me tell you here, I have not used the word that Buddhism is reducing in India. I am telling I wanted to study the perspective of Anagarik Dharmapals on the topic of decline of Buddhism in India. Because many scholars have studied how the Buddhism is declined, being a very big religion in ancient time. And Anagarikas have a very different, distinct information or distinct opinion about decline of Buddhism, which has been studied very less among the Buddhist scholars. So this is what my aim was. Yeah.
Please.
Yeah, sure.
This decline of this particular Buddhism is also very much connected to the decline of the university system. Right?
University system, yeah.
Right? That's a connection I think we need to learn from that.
Yeah, very true. Yeah. Because Buddhism, there is a concept called vihara. The vihara concept is related with the university and institutions, not with the religious, rituals and all. So it is very true what you are saying. Yeah.
Great. Thank you, sir.
Yeah, thank you so much. Thank you. Thank you.
Thank you, Amit. Next, we have Miss Radha Iyer.
मैं ऐसा करूँ कि आप करो because I don't know. You move it, no? You look here. Thank you, sir. You have. Oh. Good afternoon, everyone. First, let me thank Hindra Vaidande, sir, Nilesh Wak, my sir Rahul Prakash, sir, Dr. Rahul Prakash, then our director, Savita Moge madam. I am from Maya Somaya School of Music and Performing Arts. I am doing PhD research under the guidance of Dr. Meeta Pandit. And my topic is, everyone is familiar, revival of Indian folk music and adaptation of traditional folk music in Indian films. You might be thinking nothing new, but mine will be unique. Because I am one of the few who got the opportunity to take award from Lata Mangeshkar, Bhimsen Joshi, and my favorite Bismillah Khan Sahab, Ustad Bismillah Khan Sahab. So from each one I have collected the data. You will never find anywhere. And the topic I have, first of all, let me tell you, the culture and diversity of India is reflected. It has not come. This is my topic. 1 minute, revival, research of introduction. I will. Okay. My research objective is here. I have selected 4 states. Since I stay in Mumbai, I have selected Maharashtra, then Uttar Pradesh, Madhya Pradesh, and Bihar. The methodology is, in order to fulfill the research objectives, this study adopts explanatory research approach, which includes collection of secondary data. I would like to collect data from you people also, because many of are related with industry, in the film industry. Okay. From several published books also I have taken articles, publications, encyclopedias, magazine, everywhere. But I will see that it is not repeated. Mine will be unique, as I told you. Okay. The objective is, as I told you, I have selected 4 states. The conclusion is that to preserve our folk music, and as everyone knows, in Maharashtra, Ganpati is, we all worship. In Mauritians, they worship. So I will be giving in my book each and every bhajan with time, with rag, with translation, if it is in Sanskrit. And this conclusion is that, 1 minute, sir. The conclusion is that the traditional of folk music consists of rich blend of indigenous knowledge which have been on decline in the day and age. So to preserve that, and this study will mainly focus on revival of traditional folk music in mainstream of Indian music industry, which is somehow losing its charm. So without your cooperation, I won't be able to do. I mean, I want some collect the data, rare data. Otherwise, you will think that is there, this is repeated. Mine, not even a single will be repeated. And I have already forwarded my photographs with all the Bharat Ratnas to my director, madam, to my sir, Rahul sir also. Any questions, sir?
Madam, we understand that you are a very renowned singer.
It's not today. I have to present this right from, because some tech problem was there. It was not going.
We request you to sing one abhang or bhajan, whatever, so that we can.
Yes. I will definitely.
Very short one, please.
Definitely I will. But today,
Need music. They
Because
can
my
put the
name
music
now
also.
just now included, I was fully tense right from the morning.
I think singing will make it lighter, ma'am.
For de-stressing, that will help you.
Yes. Just 2 lines.
हेरी मैं तो प्रेम दीवानी मेरा दर्द न जाने कोई ना मैं जानूं आरती बंदन ना पूजा की रीत है अनजानी दरस दीवानी मेरी ये पागल प्रेम लिए री मैंने दो नैनों के दीप जले पिरोए हेरी मैं तो प्रेम दीवानी मेरा दर्द न जाने कोई आशाओं के फूल की माला सांसों में संगीत इन पर पोली चली री जानी अपने मन का मीत लिए री मैंने नैन डोर में सपने लिए पिरो ये री मैं तो प्रेम दीवानी मेरा दर्द न जाने को.
Thank you.
Fantastic. Thank you, ma'am. Thank you.
Now my
आपका दर्द हम
tension is
जानते हैं, ma'am.
finished.
We know your dard. You will be the best. Don't worry.
Oh, thank you. No, I'm tension से free हो गई है. Thank you. Thank you, Vaidyanti sir.
Thank you, Radha ma'am. Next, we have Gaurang Shetty. That's the last presentation we have for the day.
प्रेजेंटेशन मोड बन जाएगा. स्क्रॉल करना पड़ेगा फुल स्क्रीन. Thank you. Yeah. So thank you, ma'am, for setting up the mahol. I hope after my presentation, everyone will still be in a good mood and yeah. So they will give a good feedback. So just to give a context before the presentation, hi, I'm Gaurang Shetty, and like my guide is Dr. Radha here and Dr. Prema, ma'am. So I've been in this space. So the research which I'm doing, I've been working in the space since last 14 years, and the space is basically startups and in business incubator. And so in the last 14 years, a lot of things have happened in the startup and business incubation space, wherein the government has taken a lot of initiatives to drive this this entire industry. So business incubator as an industry was there since the 1980s, 1990s, but since 2016 it has caught up in the country, and a lot of states are now trying to race among each other to show the state ranking कि हमारे पास इतने business incubator है, हमारा ranking अच्छा है. Like Maharashtra was claiming that they were second now. Bangalore, Karnataka was claiming they were first. So now there is a big competition between these states, and the government has put more than 10,000 crore of budget to create more business incubators. So just to give you numbers, so before 2016 there were only a few incubators, less than 30. And now I think as per the Startup India report, so the incubators have grossed more than 1,000. And it will reach up to 10,000 incubators in the next 3 to 5 years. So that is the plan of the government. They have kept a corpus of funds to set up more and more incubation centers across the country. And specifically in Maharashtra, we were one of the top 16 incubators to get funding from MSINS, which is Maharashtra State Innovation Society. And again, Maharashtra government also has planned to set up more than 50 odd incubators. I think the number will cross 100 in the next 3 years easily. So one of the biggest problems with this entire market, with this entire industry, is that
startups to hai, incubators bhi hai, but then which incubator is doing what? And because they are using taxpayers' money, right? So 10,000 crores taxpayer ka paisa hai. And so much money is going here, and how do you know that the money is going in the right direction? Which incubator is getting the funds and what is the performance, what is the outcome? So there are few researchers who are working on this, and there are very few papers, like we can count them, less than 70 papers as of today in this space, which are actually gauging performance and which actually have some defined metrics to gauge the performance of incubators. So that's why this topic, the performance framework for business incubators in Maharashtra's higher educational institutes. And there is a circular from Ministry of HRD that like all higher educational institutes have now mandate they have to set up business incubators in their institute. So that's why this is very significant. And many of these new researchers like Rathod Agarwal, Hellman, so they have also pointed this out very correctly that the models of incubation also is changing. Like initially, incubation centers were only a brick and mortar center, but now the models are evolving, and that's why it's more important to understand these models first and also evaluate the performance based on the models that they are proposing. Like now there is a virtual incubation model. Startup can sit in Bangalore and still be associated with us in Somaya, and they can work with us. But then to evaluate that interaction, to evaluate that performance, there was no parameter defined before, but now there are researchers who are figuring out and pointing out new ways. Like the intake of the startups also has a lot of parameters like selection performance, then financial viability. So all these parameters have been like there have been studies, research on these parameters. So the objectives and the outcome specifically was very clear after reading these researchers' work. So to determine success outcomes of government-funded business incubator hosted in higher educational institutes in Maharashtra, and to determine factors that lead to grant support of business incubators hosted in higher educational institutes in Maharashtra. So the second objective again is very important because many incubators are getting money from the government, but the government support is only for 5 years. What of that? So after 5 years, you have to sustain yourself. So you need to have exits. So incubators typically take shares in the startups. So kuch exit hona chahiye, incubator sustain hona chahiye, because you are building so much of footprint. So for example, IIT Bombay has say 75,000 square feet. TIA Best 1 million square feet of space. So to sustain that space, they also need to generate revenue and to also plow back to their own institute. So these are the hypotheses. So just I'll read a few, like selection performance is positively related to the business incubator performance. Startups target market is positively related to the business incubator performance. So I will be using a mixed methodology approach because like the interaction, so most of the researchers have only collected data from startups. There are very few researchers, like again less than 10, who have actually gone ahead and spoken to the business incubator managers, and they are trying to collect data. So in fact, there are few researchers in this space who have tried to do this, but they have not found like meaningful insights. So that is the area which I want to explore. I want to get insights, data from the business incubators ecosystem, the team behind it, the management, and yeah. So both the approaches would. Yeah.
Very interesting topic, but I think we have time restriction.
Yeah. Thank you.
Too good. Any questions? Yeah.
An organogram for the incubator here. You are running an incubator here. You are heading it. So do you have some sort of an organogram?
Yes.
So how does it function? Like, you know, what is it that you do? So, because I see business incubation. What actually you are incubating is, you know, some technology and products, right, which you need to develop to do business.
Yeah.
So I think that technology word is critically missing.
Yeah.
So, like, what is the mode of operation? Like, there are many scientists and you know, technologists in the campus. Like, you know, are you reaching out to them? And you know, what is it that you are doing?
So there are different models for incubators. So our model is like we have 3 buckets. The first bucket is the community bucket. The second bucket is the lab infrastructure bucket, and the third bucket is the incubation bucket. So incubation always has a pre-incubation program. So for example, if you have an idea, you might not be ready with the prototype. You might just have filed a patent because you have some insights and you have done some research work and you have published a paper. But then the prototype is very important to get that seed fund, right? So there are different grants based on these each stage. So like TRL level, there are different TRL levels. So based on these TRL levels, we have created these 3 buckets. So the first community bucket actually helps us help the innovator interact with like-minded people so that he can validate his idea, go and speak, get feedback, also meet different set of customers through that community bucket. Second bucket is lab bucket, wherein once he has some validation, then you can go use the lab and build resources. Nowadays, many of these government-funded incubators have labs which are free to access. So you don't need to spend any money to use these machines. You can directly go and use the machines, and you can start rapid prototyping. And once you have a minimum viable product, you can apply for government funds, and then you can go into the incubation program, wherein Nidhi Prayas is, for example, a 10 lakh grant, wherein you can get the 10 lakh rupees, and then you can build your prototype, and then you can validate it and present it to a committee, and then you can get the next round of funding. Yes. So the Nidhi Prayas program, there is a 10 lakh grant, then there is a EIR program, Entrepreneurs in Residence program. There are n number of such programs listed on government websites. So our job as a business incubator or a technology business incubator or a BIRAC funded incubator is we have to list all these programs on the website so that any innovator from the university or the higher education institute can come and access to these funds or grants. Yes. Yeah, so we also won the national award for our work in the incubation space, so we would love to host you and show you the program.
Is the process what you are mentioned on your website, Lidl website?
Yes. So all the grant programs are mentioned, and each of these grant programs have...
And how to get that?
Yeah.
Is it mentioned?
Yeah. Selection process, eligibility criteria, everything is displayed.
Good.
Like today is the deadline for the Nidhi Prayas grant, which I was mentioning. So we have more than 280 applications already. So these are innovators from different institutes across India who have applied for the grant.
How many from our campus have applied?
We'll have to check the data, sir. So usually...
But did we do any promotion for that?
Yes, so we send out emails to everyone, email ID, students' email ID. We also reach out to the concerned departments based on the... Yeah.
So one more last question.
And on social media also it is there. On all pages usually it's there.
Want to know the reason why you are not chosen colors for making PPT?
Sir, everyone does it, so I like to give it very clean.
No, but brand guidelines was required.
Yeah, sorry, I missed that.
Sir, the chairman, his presentation should have been at the beginning only.
Yes.
Because we are talking about transnational research, interdisciplinary research. People have proof of concept ideas, some products. So now they should connect you, how they should connect, what are the scope, what are the funding possibilities, opportunities, and who will help. All that should have been very lucidly, simple language you should have explained in the beginning only. And many entrepreneurs are ready behind to invest and take forward their ideas.
Yeah. So, sure, sir, sure. So just to give an example, आपने Zepto use किया है?
Yes.
Order किया होगा कुछ तो Zepto का first interface अपने students नहीं बनाया, सोमया के students नहीं बनाया, but जावेद खत्री and मंदार सावंत. And that product is being widely used now by everyone, right? So Zepto founders actually approached them to build the first interface for them. Like PharmEasy's first interface was built by our team. So we have the capability to build world-class products here in Ghatkapur and also ship products globally. Yeah, the Square of product also was built here. So it's the world's smartest chessboard, and it is shipped to more than 100 countries now. So yeah, there is potential in India. That's why all these government schemes are popping up, and many people are getting benefit from it.
You can urge everybody to contact you with the...
Yeah, please do reach out to me. I'll just share my...
And Gaurang, your objective on this is something that you're talking about evaluating, what people have been doing it, right? People have only done 70 papers, that by the count of it.
Yeah.
So do you think if you need to increase that, you are the key people?
So there are researchers who are now publishing papers and exploring this area because there is a pressure from most of these governments. Like you see the advancements in AI, a lot of researchers are now pushing papers in these disciplines, and many of these research typically happens in universities like Stanford. There is if you go to Stanford website, there is a Stanford AI engine now which is available to like ChatGPT only. So the university also is driving this through their incubation program, to their research, to their startup programs, and they want to spin out such entrepreneurs. Like Google was a product of the Stanford incubation ecosystem only. So we also want to do that here.
Fantastic. Good luck.
Thank you, sir.
Thank you.
We have today's last presentation by Kamalika Ray.
I think we all enjoyed the last performance a lot. Thank you, ma'am.
Good afternoon, everyone. I promise I'm going to wrap up early. Sorry for my broken voice due to ill health. My name is Kamalika Ray. I am doing my PhD in the discipline of Commerce and Business Studies, and my guide is Dr. Sachin Achrekar, sir. Uh, So my title is the study of the impact of CSR, that is corporate social responsibility, on the financial performance of selected NSE listed companies. Why this study is important? Why? Because inherently, whenever we are thinking of doing any good, we are generally not asking for anything in return. But when we are talking about commercial businesses, we are trying to make it more formal, and we are definitely trying to think of it as a business strategy, if not a profit-making initiative. So CSR right now is a very important, critical to business sustainability, business performances. Also nowadays, all of us, the prospective, the existing investors, we are now moving towards responsible investing. We are all aware of the concepts of greenwashing. We all are trying to do good for the planet as a whole. So that is why it is very important to see and measure whether doing good is actually bringing anything back in monetary terms for the companies that are listed on NSE, and they are actually doing the maximum trade in our country. So that was the reason why I have chosen the NSE listed companies. The problem statement, the main problem statement that I was trying to understand is there at all any effect of the CSR expenditure on the financial performance? There are many other sub-questions that depends on the industry that the company belongs to, the nature and the age of the industry that we are talking about, or the company. So research objectives, of course, is to first ascertain and trying to understand what kind of CSR is being done by the major companies in the country. And when I mean the major companies in the country is the Nifty 500 is my population. Of that Nifty 550 companies that I'm looking at, because there I'm also getting the data very easily available, publicly available data is there, both to the perspective of the financial performance and the CSR. The next is, of course, to examine the financial performance. When I'm doing these two, CSR expenditure calculation and the financial performance matrices calculation, I'm going back to 10 years. So I'm looking at a decade performance. So I can have a whole trajectory of the journey of the CSR and how the decision making also may be changed of the CSR committees of these respective companies. Thereafter, the third objective is to try to find a correlation between them and find, of course, what is the impact of these CSR. The fourth is I'm trying to find the influence of a moderating factor, that is age also influencing these decision making, and is age also influencing the relationship of these both. Methodology I have majorly based on the secondary data that is available publicly. I am also validating these data firstly from the annual reports from the company websites. Also whatever data is available from the reputed depositories of Bloomberg, Provis, and then validating with the national portal of CSR as well. So the expected outcomes, of course, I'm going to try to think positively that there is a positive effect of CSR expenditures, because that is going to help build up your brand reputation, your brand value, and also in the long run, it is helping us to achieve the UN Sustainability Agenda of 2030. So that's the expected outcome, and I'm open to questions if any.
Outcome-wise, I would say CSR majorly has been used for company sports.
Yes. So right now my data collection is going on, sir. So I am seeing that there is a difference in as per the industry that the companies belong to. So sometimes the major focus is on education, sometimes it is on sports, but majorly the good thing about this is almost all the companies that I have collected the data on right now, they are focusing on rural development as well. So that is also something which is coming out very clearly.
I think your finding will help us, because mostly the impression is that it goes to the sport, like Geo would spend entire money on IPL,
Right.
which cannot be using your own money for
Correct.
your
And so
promotion.
I'm also trying to find out whether at all there is any difference in the decision making in between the industries, so that they can help the CSR committees also to better their decision making, because there is no standardized CSR metric as such. And we are just depending on we do not have any indices also to measure the CSR. ESG, we have Sustainalytics is there, Morningstar, but CSR, there is no focused index as such now.
Exactly. The impact will come to know once you've done the research.
Yes.
Great for that. Thank you.
Yes. Thank
Anybody
you so
else?
much. Thank you so much. Thank you.
Okay, thank you, participants. In this session, total 16 presentations has been done. This session is most interesting session, as thank you the UG students as well as Radha ma'am for making this session very interesting. Now I request Ejaz sir for his remarks.
Thank you, sir, for allowing me to do this. First of all, my apologies that I have been late. We had a BOS meeting for the sports exercise science program, and members have come from all over the country. So I could not pardon that, and I have sent my email, which was too late in the night, that I would be here at 2:30, but I managed to be here at 2 o'clock. Entertainment-wise, Radha ma'am, it was too good. आपने हमारा रिक्वेस्ट लिया और बहुत ही सुंदर गाया आपने. नहीं, ये लोग बजा रहे थे बजर, मैं तो सिर्फ देख रहा था. It was a great experience for me to listen to this, and I think Nitin sir's, Dr. Nitin's remark was fantastic, that we should have started with Gaurang, where we could understand how we getting into it and what are the different options that's available for all, all our candidates who are here. Especially when I heard that the engineering department is getting into humanity, that was something a great experience for me to understand multidisciplinary, and this is what we are looking forward to. And the icing on the cake was, of course, undergrad. They are really doing well, and this is what the whole idea is about this conference. And I wish all the best to everyone. In BSc Sports also, we are trying to make sure the same thing, where the undergrads are taking part in doing research, and these are the future for us. Thank you so much.
Good evening, and it was truly a remarkable session with so many surprises for us. I request, it is an honor to have Dr. Lisa Tiemann, an associate professor at the Michigan State University with us. May I request Dr. Tiemann to kindly come on stage and felicitate our chair, Mr. Azaz Khan. Just a second. Yeah. Dr. Lisa is an associate professor in the Soil Biology and Ecology Department of Plant, Soil, and Microbial Sciences. Michigan State University. Thank you, Dr. Tiemann. Thank you. Sure. Sure. Yeah. Okay.
It's actually my privilege and pleasure to be here today, and I'm sorry I couldn't be here for longer. I was actually up working with school kids in Nareshwadi this last week, but it was good to see a few of the presentations and some of the really great work that's going on here. And yeah, I'm excited to see what the future holds.